All right, I think our participant list is slowed down, Courtney. Would you like me to walk through these?
Sure, I can do that if you'd like.
That'd be great.
All right, thank you everyone for joining us for today's webinar series. Today we will be focused on research development and other opportunities. But as you can see from the schedule, there were several other webinars that were given. If you're interested in them, they are posted online and you can always go back on the YouTube channel and see them. And for the next week, we will go through the stepping stone opportunities. Next slide, please. And then we just want to give a thank you to all of you who provided questions for the panelists as part of this registration. However, if you do have a question while we're going through and asking or hearing some answers from our panelists, please use the Q&A box and present your questions that way. We will do our best to run through them and to try to have our panelists answer your questions. You can also vote for questions you would like to see addressed as well. Next slide, please. And then we will go through a quick poll just to get some information from you, the audience. Great, and it should be popped up on your screen now for your poll and just asking you what your current status is if you are a student. So if you can begin to add in those answers, that will be really great.
And it looks like our first poll is closed and the majority of folks on today are undergraduate students. So I think we can move on to our second one.
Awesome. And just for you all, just answering how familiar you are with clinical genomics research and development careers. This will be helpful to get an idea. And hopefully most of you will say you may not be as familiar and you're going to learn today. Great. Well, thank you all for taking those questions. I do want to take a moment to introduce you to our panelists today. We have Nara Sabrera here. We have Nicole Vasilevskiy, Adriana Mayero, Alex Wagner, Neetu Shah, and Orion Busky. Welcome everyone, and thank you for coming today and being a part of this workshop. So we'll take a few minutes now as we work through the rest of our workshop here, just to have everyone give a chance of about 5 minutes to introduce yourself, tell us who you are again, maybe where you went to school, what's your current career that you're in, and just what you do. Obviously, if any of you have a similar background as others, you can kind of chime in to say you had a similar career path, but certainly highlighting anything that is unique about yourself. Maybe we'll start with Adriana.
Hi, good morning, everybody. So I am a certified genetic counselor, but at the end, I work at the NIH, where my official title is staff scientist. Within the NIH, it's divided into different departments. I'm at the National Library of Medicine in a department called National Center for Biotechnology Information, or NCBI. And I have been at NCBI since 2010. Currently, I direct the NIH Genetic Testing Registry, or GTR. And this is a database of labs and genetic tests that can be clinical and research. And it's supposed to be a resource for clinicians. So we get the labs to register that test information, and then health providers can search for the test that they need. And then we supplement data with disease information and gene information for molecular tests and practice guidelines and things like that hopefully help the genetic counselor, geneticist, or nurse or another healthcare professional that needs to select a genetic test to learn about the test and what the test is for. I do want to say that for GTR is a genetic, it started as a genetic database, but actually recently expanded its scope to include some infectious disease tests, especially for COVID-19. And I just want to say that you can always leverage your earlier academic career and professional experience to kind of help the field or the job that you have at the moment, even though it's not what you specialized on. So in my case, I specialized as a genetic counselor, but I am now, including infectious disease and COVID-19 tests into a resource. So right now, I also direct a portal called MedGen. So it's a website with all of the genetic phenotypes that come from different authoritative sources. And we also have the medical genetics summaries, which is a separate resource that focuses on pharmacogenetics. So it's articles that synthesize all of the pharmacogenomic evidence that's out there in the community into practical information that can be used at point of care. Now, I started, I did my undergrad at Stony Brook University in New York, and I graduated with 2 degrees, one in biology with a genetics track, because at that time there was no genetic specific diploma. And I also did clinical laboratory sciences. So I graduated as a medical technologist as well. And this is actually what I used to support myself during my graduate school. Now, during rotations, I had to ask for an extra rotation in a genetics lab because that was not part of the curriculum. But that increased my exposure in genetics. And just to say, being a medical technologist, part of it is microbiology. I had to learn and rotate through microbiology, which actually right now does help GTR help the community with these COVID tests. So, it's something that I could leverage from my background. So how I learned about genetic counseling, it was actually during a developmental biology class. I was just chatting with one of my classmates that was thinking about genetic counseling, and that's by pure chance how I actually learned about genetic counseling as a career. And then I actually looked around to see if there were any genetic counselors, but Stony Brook is kind of in a rural area of New York, so there weren't actually any genetic counselors around. So I did a lot of the research myself about the profession. And I actually enrolled in my master's course, before I actually talked with a real genetic counselor. And it wasn't until I started my graduate studies that I actually talked with genetic counselors. Okay, then I did my master's, and after my master's, I actually started working at the Howard University Department of Pediatrics and Child Health. And at that time and for that job, I did wear many hats. So I wasn't just doing genetic counseling, even though that was a big part of it. And the majority of the patients were in hemoglobinopathies. But aside from the genetic counseling, your traditional genetic counseling role, I was also a research assistant, or I started as a research assistant for the pharmacogenomics of morphine in sickle cell patients, and then kind of graduated into a research coordinator for different programs that were assessing the clinical progress of pediatric patients with hemoglobinopathies, especially sickle cell disease. And then I also served as a program manager to improve access to care services for children with hemoglobinopathies in DC. And I was both a case manager and a project coordinator for this project, where I basically did some social working, outreach, advocacy for patients, community screening, blood drives, you name it, I was on it. And this was sponsored by HRSA and the DC Department of Health. So there was a lot of grant writing to keep that going. After all of this, or while I was doing all of this, I did see this job at NIH for GTR, where I work right now. And at that time, GTR was being developed, so it wasn't even public yet when I came on board. Now, NCBI is awesome, and they have amazing bioinformatics skills, but they have no one to give the clinical experience. Or actually, there was one genetic counselor that had been brought in to give the clinical experience, and they kind of needed some more perspective. So that's how I came in. So I was the second genetic counselor to come on board. And then MedGen was born, and the medical genetic summaries were born. And I was a curator dealing with content, dealing with submitters, giving us data, users using the data, telling the community how to use us. And when our director left, I kind of stepped up and took this leadership role, and that's what I am right now.
Oh, that's great. Thank you so much, Adriana, just to hear the journey. And I think it's really nice to what you're saying, right? You may start in one area, but if you have different opportunities, you can kind of expand and gain new expertise as you move through. Thank you. Nara, you want to go next?
Yeah, thank you for having me here today, and good morning, everybody. So I'm actually a clinical geneticist. I did medical school in Brazil, and then I did a residency in clinical genetics in Brazil. And then I came to US, I came to Hopkins in 2007 after I finished the residency to do a PhD. And then my goal was to go back to Brazil, but with time, as the work progressed through my PhD, I decided to stay in US and be able to work as a physician. I had to do the steps, first of all. So if you do medical school in another country, you have to take the boards here in US. So I had to go through that. And I did that towards the end of my PhD. And then when I finished the PhD, I was still, so there are 3 steps, 1, 2, and 3. And I think I finished the PhD. I had finished 2 of them, but then I did a postdoc, and during the postdoc, I finished the 3rd one. And then I could be a physician, but I also needed to be board certified in clinical genetics in US. To do that, I actually had to repeat the residency. They gave me a few years of credit because of the residency that I had done in Brazil and the PhD that I had done here at Hopkins. So I needed to do 2 years of clinical fellowship. So I did that. And after that, I was hired at Hopkins, and I'm now assistant professor at Hopkins working with clinical genetics and research genetics. So my research is pretty much focused on trying to understand what's the genetic cause of the diseases that I see in clinic. So we use different methodologies, and I don't know if you have heard of any of them, but the main one is whole exome sequencing, now more whole genome sequencing, but newer things like long-read genome sequencing, RNA-seq, metabolomics, proteomics, methylation. Any methodology that's out there, we try to bring into our research to find the genetic cause of the disease that we see in clinic. There are a few diseases that I focus more in the lab work that we do in my lab, trying to really understand how not only what genes cause the disease, but how they cause the disease and how we can use some kind of medication to stop the disease from progression or making the disease to regress. So I think very quickly, this is what I went through. I don't know if I'm supposed to go through all the other questions now, or you were doing that later, Courtney?
We'll go through them a little later and have everyone, but thank you, Nara. That's interesting aspect too, to bring in being international and, the steps that might be additional to think about for those individuals who may decide to stay in the US and come with different degrees. So thank you for providing that.
Of course.
And we'll move on to Nicole Vasilevskiy.
Hi, I'm Nicole Vasilevskiy. I'm with the University of Colorado, the Anschutz Medical Campus. I actually just recently moved there from Oregon Health and Science University, where I've been for the past 10 plus years. And my background is a bit different than the others on this panel. So I am trained as a biologist in cell and molecular biology. My undergrad degree is from Reed College in Portland, and I have a biology degree, and my PhD is from Oregon Health and Science University here in Portland, which is where I still am. I'm working remotely now to the University of Colorado. And I had that kind of a traditional basic science training where I worked on the bench in molecular biology and immunology. And then when I graduated from graduate school, I no longer wanted to be on the bench and doing wet lab research. So I was looking at other career opportunities that were non-traditional for the type of training that I had. And I ended up getting a job at OHSU working as a biocurator. And that's what I do now. I'm a biocurator for a group at Colorado. It's the Translational and Integrative Sciences Lab. So what I do is I do curation and ontology development for biomedical ontologies. So biocuration as a field is standardizing and structuring data to make it more computational and interoperable. So as biocurators, we annotate data sets and standardize data and using ontologies. And ontologies are standardized terminologies that they're like a taxonomy or a controlled vocabulary, but something that's unique about ontologies is that they have logic built into them. So you can do kind of sophisticated data analyses and computational analyses using ontologies. So a lot of the work I do is building ontologies, and one in particular is the Mondo disease ontology, which is a classification of diseases across different species, and it's very widely used by ClinGen and other groups for standardizing genomic data. And this enables us to standardize the data and make it more computational and do downstream analyses on it. So yeah, so my career path is a little bit different than others, and the field of biocuration is relatively small, but it's been a very cool career path, and I really enjoy it. So I'm excited to talk to you all about it more today.
Oh, thank you so much, Nicole. I like the fact, just showing the ability to adapt and, change, and many of you, right, how you start as one and maybe find different areas when you go through science. So that's great.
Yeah.
Let's go on to Orion.
Thanks. And thanks for having me and giving me an opportunity to talk to everyone today. Yeah, so I think let me think about what the right way to present a summary is of how I found myself here. But I guess to start with, I run a software company based out of Toronto, Canada. We make a software tool called Phenotypes that's used in genetics clinics and hospitals and health systems for managing a lot of the clinical information that's necessary for seeing patients and helping to diagnose rare genetic conditions and interpret the patient's genome using a lot of the terminologies that Nicole and others help curate that are absolutely essential for making clinical data computable in genetics. So thank you, as always. So I guess I found my way here on the industry side through a bit of a longer process going through academia, actually. So I'm originally from the US, out west in Washington State. I wanted to be an architectural engineer. And so until I took one of the mandatory classes in biology, and in particular in genetics, the high school that I went to was shockingly well equipped for sort of a not particularly well-funded high school, in the sense that we had a bio teacher who had a whole bunch of friends at pharma companies and others, and so got all the hand-me-down and expired reagents and things like that. So we had a single capillary DNA sequencer in our high school for no otherwise good reason, and thought that was amazing. And I absolutely wanted to go into genetics for my undergrad. And so I ended up going in-state into the University of Washington, and they have a genetics and genome sciences department, but there was no undergrad program. So the closest thing from my perspective was going into bioengineering, where there was the opportunity to work on genetics. So I was surrounded by a lot of other pre-med folks who were doing that as in training for med school. But one of the requirements that I had to take was a computer science class. So I thought I would get that out of the way early on as a requirement, and suddenly took it and was like, oh, this is really interesting. I want to do more of that. And they make you at some point major in order to keep taking classes in it. And so I double majored in computer engineering there with bioengineering, and sort of from both of those found this intersection that's computational biology, which is how do we use computers to understand biology, in particular with a focus on genetics. I got to work on a big collaborative international project called the Encode Project after I graduated undergrad for a year while I thought about going to grad school. I think for me, one of the longer term focuses was an interest in teaching, particularly at a university level. So I think the PhD was on the horizon for me. And I ended up interviewing at a whole bunch of different universities for graduate school, and found that the University of Toronto here in Canada, actually going across the border to the north, which is not the usual flow. Usually a lot of people in Canada go to the States for school, but going to Toronto ended up being an amazing fit. And I think for a lot of my career, that decision to come here ended up influencing a whole lot of things. And it's just been an amazing opportunity to have really strong computer science program at the university, right next to like 9 hospitals with research centers within walking distance. So that really paved the way for the work that I was focused on, being really at this translational genetics, computational biology space. How do we use computers to basically directly help the healthcare providers who are in the trenches and of actually helping people? How do we use that computational technology to increase the leverage for their ability to care for people? And that research that I was working on there, there were a number of projects, and phenotypes was one of those projects that then spun out. So I actually continued the PhD working on algorithms, focusing on a number of other projects like the Matchmaker Exchange with NARA, and starting to connect with the Global Alliance for Genomics and Health, and figuring out standards and data interoperability. And then after completing the PhD and postdoc in that space of sort of rare disease diagnosis and software and algorithms, I came out and joined the company, and have been running it as CEO for the last 3 years while we scale things up and now try to sort of get software into the actual hands of healthcare providers in the real world, outside of the research setting, sort of anticipating this shift of genetics and genomics into mainstreaming, and trying to provide the tools and the business cases for making that happen.
Oh, thanks so much, Orion. That's interesting. You're bringing that perspective of the intersection of computers and biology, the same for you and Nicole, and how we can facilitate computers helping us in the future. So that's really awesome. How about you, Alex?
Yeah, happy to go. Thank you for inviting me to participate on this panel. And it's really great going after Nicole and Orion, whom I know from many of our collaborative works in this space, because I too am involved in how we do kind of standardization and computability of knowledge in order to apply that to clinical care. So to give you a sense about who I am and where I am, I'm a tenure track assistant professor at Nationwide Children's Hospital and The Ohio State University. And today I'm doing medical informatics research. I'm applying my research to improve outcomes in pediatric patients. So across the street from my office, the primary hospital building intakes patients that come in with different rare disorders, including pediatric cancers. And what we need to do is identify, how do we treat these kids? How do we identify their prognosis, their diagnosis? What are the best, therapeutics we can give them, that sort of thing. And to do that, we are building computational tools that allow us to collect evidence from the biomedical literature, that use really nice upstream tools like phenotypes and standardized vocabularies like Mondo, and try and be able to automatically search all of that biomedical knowledge to help our variant scientists in our clinical team basically apply that in order to drive that diagnosis and those treatment decisions for these children. Now, to get here, I followed a by and large, very traditional academic career path. I did my undergraduate work at Iowa State University. I went there because they've got an outstanding computer engineering program. I was very excited about it, and I started there. But as I was working computer engineering, I started to gain an interest in genetics and biology. I finished, my degree in biology with a minor in mathematics. And while I was there, I also did some undergraduate research for the USDA. So for those of you that don't know much about Iowa State, and I don't blame you if you don't know much about Iowa State, it's a very heavily agricultural school, and they have a very large presence from the US Department of Agriculture there. And so I was doing research in the USDA's Agricultural Research Service, trying to help build the first soy genome at Soybase. And it was a really fun opportunity. And I know that a lot of the people on this call are undergraduates. And I just want to say, like, I really got into my career in this career path based off of, exploring this opportunity that was available as an undergraduate, just something to do alongside my coursework as a job. And I learned a lot about, building genomic assemblies, about bacterial artificial chromosomes, about, putting together, fluorescence from gels and turning that into actual sequence. And it's really exciting stuff. And it's something that I think is uniquely available to undergraduate students. There's always a need for undergraduate research on major campuses. So I would strongly recommend that the people on this call with an interest in clinical genomics see what's out there. It might not actually be something in like a clinical lab, but it might be very relevant to the work that you ultimately want to do, or might help guide you there. So anyways, after doing that for a little while, I ended up graduating. I went on to go into a traditional like 8-to-5 job. I worked at Mayo Clinic for a little. It was actually, I guess, more like a 5 to 8. I worked night shift, but basically, I was working at Mayo Clinic doing clinical laboratory work. So we were testing people for trace and heavy metals in their fluid samples, including like blood and urine and things like that. And the idea being that we're trying to detect things like elevated lead levels or aluminum toxicity, things like that. And so it was a very interesting experience working as a clinical lab tech. I did that for 2 years, and it helped me appreciate and get a perspective for the way that large medical institutions work, about the sort of team effort that you need to, and the philosophy you need to have in order to actually do like world-class clinical healthcare. And I've really taken that mentality of collaborative and team efforts along with me throughout the rest of my career, because I think that was one of the most important lessons I learned along the way. But I did eventually decide that I wanted to go off and go back into research and run my own research program. This is something I had wanted to do ever since my experience with the USDA. And when I went to do that, I was looking around for a good program, and there's this very interesting one down at the University of Iowa, not to be confused with Iowa State, where I did my undergraduate work. But the University of Iowa, which is on the east side of the state, they have a very large clinical hospital. And it's an academic hospital that has a very good research program in eye disease and eye genetics. And so I went there to go study rare heritable retinal dystrophies. So these are disorders that cause people to go blind over time, that they inherit from their parents. And when I was there, I did, it's more of my computational work. So I was in their computational genetics program. And I studied bioinformatics and computational genetics. And while I was there, I was building out some machine learning tools to help us prioritize what variants might be causative for these patients with these extremely rare and highly heterogeneous disease. I think one of the bigger challenges in clinical genetics is when you have diseases where you just don't have enough people with the disease to really help you know what the meaning of the variants are. This is, I think, one of the big open challenges in the field, and there are a lot of approaches and a lot of research going into how we can address it. And so to that end, I was doing some machine learning work to try and help figure that out. I built a couple of web tools that are, looking back, were probably not that great, but at the time I was very proud of, including the ocular tissue database. And then after that, I went off and I got my PhD, and I said, okay, well, now I really want to, I want to get into cancer. I want to do cancer research. I think, as is the case with many cancer researchers, I knew some people that were affected by the disease, and it became personally important to me to study in this area. And so I decided to transition to that during my postdoc. So I went down to Washington University in St. Louis because I clearly absolutely refused to leave the Midwest. And I went down to Washington University, and I studied with a team there, it's two twin brothers, Obi and Malachi Griffith. We're running a lab where they put together a resource called the Drug Gene Interaction Database. And I wanted to come in and help build out this tool and turn it into something usable in the clinical setting. And in 2012, there was an article about a Dr. Lucas Wortman who was suffering from a type of blood cancer that was, really knocking him out. And he went and he did some of this targeted sequencing at WashU, and they identified a largely overexpressed gene. And we used the Drug Gene Interaction Database to find, what is it that was, what could we do about this? We found some potential therapeutics, applied them, and he's still in remission to this day, now almost 10 years later. So it was a really exciting outcome. And it really helped solidify why I wanted to do this type of work. So I stayed in that postdoc for a long time. Most postdocs last 2 to 3 years. I was there for 4. And in that time, I helped build out a couple of other resources, including the Civic Knowledge Base, which is how we interpret variations in cancer, as well as I worked for this consortium, the Variant Interpretation for Cancer Consortium, building out a meta-knowledge base of resources like Civic and other ones that had popped up from a number of different private and public institutions, harmonizing that content and demonstrating why it's important to, again, work collaboratively and as a team and standardize this content, because it's such a big space and we need every hand to help do this stuff. So I took all of that work, I applied it to a research grant from the National Human Genome Research Institute. It was funded, and I took it with me to my first assistant professorship, which I started a year ago here at Nationwide Children's. And since then, I've been working on that grant, developing some of these tools I started in my postdoc, and helping to take this content and make it part of a larger community effort to standardize this type of knowledge and make it easier for laboratories around the world to use it and apply it to clinical care.
Oh, thanks, Alex, for sharing us with that journey. I think it's great for you and all of you to talk about the different ways that you've made it to where you are today. And last but certainly not least, Neetu, if you would share with us, what you do and how you got there and where you went to school.
Hi, thanks, Courtney. Hi, everyone, I'm Neetu Shah, but let me just first start by thanking for inviting me to this panel. I'm very delighted to be here today. I'm a lead programmer analyst at Baylor College of Medicine, led at the Bioinformatics Research Laboratory, led by Dr. Alex Milosavljevic. And as a lead programmer analyst, I'm a lead engineer of a couple of tools and resources provided by Baylor to ClinGen. I'm not sure if you're familiar with evidence repository or actionability curation interface and criteria specification registry. These are some of the tools and resources that Baylor provides for helping researchers curate variants, and not only curate, supporting curation of variants, also dissemination of the curated knowledge. So I am an engineer and I'm involved in providing not only coding, full-time coding, but also providing technical solutions and building resources for clinical genomics and the wider community. From the late last year or so, I have also been, have a dual role in product ownership. So that is an exciting skill set that I have attained for the past 10 months or so, where I, especially for new products like criteria specification registry, besides having that software engineering and engineering skill set, I'm kind of understanding the importance of communication, how important it is to communicate. And get the information and bring it back to a technical team and design. So I'm in a very exciting phase of my career right now. And I did my master's in computer science and bioinformatics, but there are others in the panel who had, background in biology. So I also had a master's in microbiology. And when I moved to US, I wanted to start something new. So I joined the School of Informatics, Indiana University. They have a great program, Masters of Bioinformatics, a good blend of biology as well as computer science. And it was exciting. I started learning core computer science courses, and that's how I reached here today. And I joined Dr. Milosavljevich's lab in 2013 and has been associated with ClinGen since then.
Great, thank you so much, Neetu. I just want to open it up for anyone in our panel now and ask a few questions. And any of you have any advice to give or any answers, please pipe in. But just thinking about, many of you had talked about your different careers and things like that, but what do you think made you a successful applicant when you pursued this career path? If you have to think back of how you've moved through these different areas, whether graduate school or going into your particular position, is there anything that you think maybe made you stand out or could be advice for others?
If I can say something.
Oh, yes, please.
I do think every time you think of something that you like, doing a rotation or some kind of volunteer work on that area helps you, because when you are applying for a position later or on a job or in school as a student, having some experience on the area that you're going into does help. It doesn't have to be like a paper in a very good journal. It's like any experience, knowing how to pipe it already helps you. So it puts you in a position that's a little different from someone who has no experience at all, and tells people that you are interested, that you are trying to find out about that, and that you're trying to get exposure to what you like. So I think any experience that you can get as a volunteer in a rotation, any related to what you want to do, it's valid and it's helpful.
Oh, that's great advice, Nara. Thank you. Does anyone have any other things? Nicole, yes.
Yeah, second, agreeing with what Nara said, I agree that any kind of prior experience or demonstrating interest in the field. When I got hired, actually, I didn't have any experience in biocuration, and biocuration is kind of a relatively new field. So a lot of people go from doing bench research like I did into biocuration. So it's not as common that someone would have experience, but my boss, Melissa, told me when I interviewed, I had done a lot of background reading about it. And I did background reading about ontologies, and I had like a stack of papers with me when I showed up at the interview. So she said that was one of the things that differentiated me from the other candidates, is that I actually had done some preliminary kind of work and showed a demonstrated interest. And one of my colleagues too had told me about a postdoc he had who came from some kind of other unrelated background, and he was really interested in getting into computer science, and he had kind of done like some self-taught courses, and it showed a lot of enthusiasm during the interview. And Peter said that these are the kind of people he likes to hire. He likes to hire people who don't necessarily have the experience and have the CV that shows that they have a track record of working in this field, but they have demonstrated potential and enthusiasm and willingness to learn.
Great. And I just wanted to ask one more, because we did get a question in for you specifically, Nicole, and it was, why did you decide to make the transition in your career from bench work basic science to biocurating? What was your motivation for this?
It's funny because I had never heard of biocreation when I even applied for the job. I didn't really know what I was getting myself into, but it turned out that biocreation was like the perfect job for me. I love organizing data. I'm very meticulous, and my boss describes me as I like to make the bed. I like things to be organized and neat and orderly. Bench science just wasn't for me. I wasn't super successful on the bench. I found it to be very tedious and frustrating, and I always did like kind of keeping a good lab notebook and keeping my data organized. And I feel like I was kind of lucky to find this field because I didn't really know about it going into it, but kind of serendipitously fell into it. But I made the transition because I wanted to leave the bench, and I was trying to find other career paths. And this just kind of, I became lucky and found the perfect fit for me.
Great, thanks so much,
You
Nicole.
mind if I chime in for a second?
Oh, of
I don't know
course.
how to raise my hand, so.
Oh, yes, no, go right ahead, Adriana. Thank you.
Sorry. So I just wanted to say a couple of things, and one of them is, so I also wasn't born in this country, and I had to learn certain skills and cultures, right? So one of the things that I want to say is don't be afraid to ask. I mean, I went to a school where I literally had to ask, like, okay, I took these classes in genetics. I really want the experience, and I literally like had to be the one finding the lab to do my internship and to be counted. So talk with your advisors, with your teachers, and for the most part, like people are here to help you nurture your interests and your academic career. So never be afraid to ask and even search around for the experience that you need, like the other two people have said. And the other thing that really worked for me is not being shy to try new things. So being willing to learn new skills and just not shying off from experiences. So I went to school as a genetic counselor, and right now it's so much different. So I literally lead a team of people who do databases, who do websites. So the genetic counselors like myself and other clinicians can actually access all the information they need, the tests, the diseases, what gene causes what disease, all of that information is needed at point of care. But when I went through school, it was just a given. We had the resource called Gene Reviews and OMEM, and everybody used it. Like, it never occurred to me that someone needs to do this kind of work, right? So not being afraid to really explore new skills, even though it might not, it might not occur to you, or you weren't exposed during your academic career. And now, like every so often, like I do work with Nicole, so we standardize data so people, regardless of how they need to learn about a disease, they have access to that information. So I just want to do that plug in, like, be willing to learn new skills and develop new skills during your academic career. And then once you get a job, you can expand on that.
Well, that's wonderful advice. Curiosity and enthusiasm can get you a long way for many, many different things. Does anyone else have anything they want to add? Yes, Alex.
I would just say, on the subject of, skills to think about and to maybe get some experience in. For me, of course, I run a dry lab, so we think about computational work all the time. And while it's not a strict requirement, I think a very useful skill to have, regardless of what you go into, if you're interested in research, is to get some just basic programming experience in something like Python or R. If you are able to sit down and like munch some data, you're going to get very, very far in research, because a lot of what we do is just being able to put data sets together before we can do analyses and such on them. So some basic programming skills, I think, are just a very valuable, broad purpose skill set to have in research. And so, I would strongly recommend that anyone who's looking to, maybe start a rotation in a research lab or go to graduate school or something, consider, just doing, some take-home resources on getting an introduction to Python or something.
Oh, great. Thanks, Alex. Yeah, the two sides of the coin, right? What bench creates data, computational actually reviews data on both sides. So that's really great advice. Orion, we have a question for you. How did you find the transition from academia to industry? And did you find that transition challenging?
Interesting. I have found it is certainly an interesting transition. I think it's one that I was interested in for a number of years, just trying to understand. I guess I had basically every job I had had since I was young had been on the academic side, as a researcher going through school. So it was really intrigued me, and I was interested in exploring what that other part of the economy is, or the other part of the world might look like, and just getting some experience in that. Even though my eventual goal was to end up back in academia, I thought that it would be helpful. And I think in computer science in particular, that's a really, really common career trajectory, is to go out into industry and then come back. Because I think there's something about that. I don't want to use the word real world, but like the on the ground, like outside of the particular mechanics with the way that academia works, experience that can be valuable. I mean, in some ways, there aren't as many differences as I would have expected. I think we're still in the same sort of space. It's still involving software and algorithms in this particular sector. It still involves a lot of collaborations with the same wonderful people who I was working with as a researcher. I think on the challenging side, it changes some of those relationships a little bit. Money now becomes part of a conversation in a way that it wasn't, at least when I was a grad student or a researcher on that side. So the currency is different, but a lot of those collaborations are still there. I think it makes it, I'm still trying to figure out what that relationship is, for instance, between how can I and we be in, what's the right way for us to be involved in conferences, for instance, right? I'm spending a lot more time at exhibit halls, although not the last year and a half, but then when as a researcher. So I think those relationships have changed a little bit. I'm still trying to figure out what that looks like as we go forward. But on the other hand, it does mean, and I guess a little bit is that particular to my role, but having the ability to manage and control resources for being able to do things and affect priorities on a much tighter timeline than otherwise we would be able to. Just sort of the way that granting works, I think you end up sort of oftentimes setting things out on a longer term, versus in industry, we can pivot pretty quickly and move resources around depending on where the urgency is a little bit more effectively, which can be nice.
Oops, well, thanks for that perspective, Orion. In the last few minutes, we have about 8. So please ask any questions in the question and answer if you have it. But I did want to, you've given all really great advice so far in the positive, but just also wanting to know, were there any barriers that any of you came across in pursuing your careers, or something that seemed a little bit more challenging you had to go through, and maybe what you did to be able to surpass that challenge or that barrier?
I do want to start with that.
Yes.
Sometimes people see what we have done, but they don't see how long it took and how many tries it took to us to do it. So like, just to get in medical school took me 3 tries to get into medical school. Then to get the steps. The first one wasn't the first time. The second one took me 2 times. The third one took me 3 times, and then let's go for the grants now. Just got a grant that took me 4 times. So like, nothing it's like that easy for everybody. Some people get it very easy and smooth and everything is on the first try. Great. Thank God for that. But when you see someone that achieved something or that got to a place where you would like to be, just know that it took few tries in each one of the steps that this person went through. It was not the first try. And if you try the first time and you don't get it, do not give up, because this is not how you will get anywhere. So just to let people know that one.
Oh, that's great. Persistence. Persistence is key, right? Keep your dream alive. Thank you so much, Nara. Does anyone else have anything they want to share?
I have
Sure.
a quick comment, if that's, which is, at least from, I would recommend keeping your eyes open about things that you're interested in as well, which maybe goes along hand in hand with what Nara is saying. I guess for me, it's a lot of my career trajectory has been dictated more by seeing new things that I wasn't expecting to find and going down some path there, versus I ended up in a very different place from where I thought I would be going. And so being free to follow your interests is also can be helpful.
Well, that's great. Adriana, did you have a
yeah, just to say, like, so where I am right now took a lot of like self-learning on, you know, all of these wonderful people talking about bioinformatics. I had zero at that in my academic career, and I had to learn it. And there was a huge learning curve, but it was necessary for the job that I, you know, am doing right now. So I can talk with developers, so we can have, you know, healthy products for you know, the healthcare providers to use, for the labs to use. But it did take a lot of learning, because it's, you know, like, it wasn't something that I had during the academia. So I had to really rely a lot on my team members and learning new languages, right? So thankfully I had a very supporting staff, but it did take a lot to, you know, to learn, you know, like these new huge data sets and how to manage all of these things. I think you have to be very flexible when you grow professionally, because, you know, like, I even had to learn a little, like, you know, for the development of websites and databases, you start using something called Agile. Like, I even learned that, and I'm sure you guys in the panel probably know about this, but to me, those are all new skills. So you need to be, you know, willing to do it, and then, or at least trying it out. And then if it's something that doesn't fit, you know, like, you move on to something else. But there is that activation energy, right? Like, you're going to put in positions where you're going to, you know, to have to learn new skills, and it's not all about the roses, like Nara was saying, right? Like, sometimes it takes tries and more tries, and also you have to adjust, because that's the way that our, you know, that the field is moving, right? Like, now we have all of this data. When I started, it wasn't all like this, right? So you kind of have to grow with it. And again, if it doesn't fit, there's always other options. But, you know, like, being willing to put in the effort, though, because sometimes it's a lot of work.
Well, that's great advice. And, you know, just emphasizing, as you said, just try it. And if you don't, there could be another option that fits for you. Alex, I see your hand is raised.
Yeah, I think one thing to keep in mind is that anyone who's on this call right now, you're already doing you know, one of the right things, which is getting out there, trying to learn more about what you're interested in. So you know, I think many of you, I think, already recognize the value in that. For those of you that have asked questions today, I think that that's probably one of the best things you can do in terms of, you know, overcoming obstacles, because we all face different ones. As we try and advance through our career, we all have different challenges, but a general strategy to take that's always helped me is to just go to your advisors, go to people that are in the positions that can inform you about what it is you want to know, and just be open and ask questions. You know, I think everyone on this panel here today is volunteering their time to talk to you, because, you know, I think as a community, people are very interested in helping, you know, young, enthusiastic people that want to go into this career, to help them achieve that. So I would say, just, you know, always feel encouraged to reach out and ask your questions. And, you know, the worst thing that can happen is they say, you know, I don't have an answer to give you.
Right. No, that's great. And then you can always go to the next person till you find an answer, right? Thank you so much, Alex. And Neetu?
Thanks, Courtney. If there are any students who would like to pursue career in software engineering, I would like to tell them, like, the challenges are the same as others face in their, you know, professions, but willingness to be a constant student. Like you have to be abreast of the new technologies coming in. Doesn't matter if you have good programming skills, that is not going to help you unless you know what is the current, technology, what could be the solution. A solution that was 5 years ago, which was effective, may not be the right one today. So why? Learn about it. So that willingness to be a constant student, regardless of what career you are in, it's very much applicable to software engineering. And of course, it's a teamwork, as Alex mentioned. Asking the right questions may not be right always. So talking, brainstorming with your team members, other experts in the field, reaching out to them is always very important.
Great. Well, we are right at the hour. So I just want to thank each of you for volunteering and spending your time to help, as Alex said, just provide different perspectives and being somewhat of mentors to these undergrads and graduate students as they're moving into interest in genetics or even computational biology. So thank you all very much. And we wish you all good luck in your futures and have a wonderful day.
Thank you, Courtney.
Thank you.
Thank you.
Thank you.
Okay. Bye.
Have a good day.